am just so sad we can't finish this activity together in class. So I prepared this little video to just sort of help you through the rest of this activity that we started last time. So remember, we were talking about the nephron of the kidney. So this is the nephron, and we're going to talk about diabetes and diabetic people. But I just want to kind of orient you to this picture. So this is going like, let's see if I can draw it. If this were a kidney, right, and this is going down into your bladder, then these little loops of Henle are all over in here. And so the outside is the cortex, and then the inner part is the outer medulla, and then the very inner part is the inner medulla. So that's what we're looking at. We're just zooming into one of those little pieces. And so if we look at it, I'm going to erase that. If we have, as far as concentration goes, so at the cortex, there's 300 is the concentration. So I'm just going to represent it by like dots like this, right? And then as you go down, then it gets into 400, we're getting a little more concentrated, and then 600, we're getting a little more concentrated, and then 900, we're getting really concentrated, and then by the time you get to the bottom, it's like super solid concentration, right? So we've got this concentration gradient going down. And so what normally happens is the blood enters into the glomerulus, and it's usually either isotonic, well, yeah, it's isotonic, it's at 300 milliosmol to the surroundings, and so nothing's going on. And then it travels down the tube, right, and then back up the tube, and then back down. That's the way that the fluid coming in from the blood flows. And so, and then it indicated that from A to B, so on this side of the loop, it allows water to flow through easily, but from this side of the loop, it only allows like sodium and chlorine, like ions, salt ions, to flow through. And then it didn't tell you this, but the last part of the loop is also for water, which is a really cool setup. So normally what's going on is as the blood is flowing down, where's my little pointer? Oh, it's a lightsaber. Oh, funny. Let's grab a dot. So as the blood is going down through here, it's starting out 300, 300, so they're equal, but now it's 300, and it's traveling into more concentrated area, but it's still 300. So where's water going to go? Well, normally water would start flowing out of the tube to try to make it more concentrated in the tube, because it's less concentrated in the inside, so you're going to get water flowing out like this. And then, as it flows back up the tube, it's now, by the time it gets down here, it's 1200. And as it starts going back up, now it's more concentrated than its surroundings, but water can't flow in, only salt can flow out. So what you end up having is the salts are trying to make it less concentrated, and so they're flowing out during this part. And then once we get back up to the top again, up here, we're isotonic again, and then we start flowing down, and we're less concentrated than the outside, and so again we're going to start sucking water out, and this is what concentrates your urine. So by the time it comes out the bottom, it's as concentrated as it can be and still flow. Okay, so that's what's going on. And so let's look at let's just leave this. All right, so if we're looking at a non-diabetic person, it's coming in at 300 milliosmolar, which is isotonic to what we are here. Okay, so that was number 10. Number 11, as we're flowing down, then your water should be flowing out of the tube, right? But Peter, his filtrate is coming in at 1600 milliosmolar. So he is way, way, way too concentrated in the loop. And so instead of water flowing out, water begins to flow in, right? And then here, salts are going to try to flow out. But then again, we're way too concentrated, so water keeps flowing in. So what you end up with is really, really dilute urine and tons of it. So that's one of the symptoms of diabetes, is that they pee a lot. All right. So that hopefully that helps you orient yourself to the nephron. Okay, so questions 14, 15, and 16 are talking about the treatment of diabetes. So let me orient you to a cell. So here's like a cell that's somewhere in your body, and then you've got like a capillary running by, right? So here's your blood cell, or your bloodstream, with your little blood cells coming through. Okay, and so your blood cells are traveling through, and they're bringing with them glucose. I'm just going to call it glue. Okay, and there's like no glucose in the cell, but there's tons of glucose outside of the cell. And so what needs to happen is the glucose needs to go in, but it's too big to go in. And so we have this little channel called GLUT4 that will open up and allow the glucose to flow in. But in order for the channel to be there on the surface, we have to have an insulin bind. And the insulin is what allows, it recruits this GLUT4 protein to the surface. So if you don't make insulin, if insulin is gone, then this protein never comes to the surface and glucose can't get in. Okay, and so what we do for diabetics is we give them shots of insulin so that it will bind to it and allow the glucose to flow in. But if you give them too much insulin, then every cell in the body will take in the glucose, and then there'll be no glucose in the blood, and people will begin to starve. Their brain cells will start to starve. But number 15 says, if you have type 2 diabetes, it's not that you don't make insulin. You have insulin, it's just that it doesn't respond to the cells. So even though the insulin binds, these receptors don't come to the surface. So what we have to do is get food into the cell a different way, and it turns out that proteins and fats and things go through a different channel. But if you are on insulin therapy and those channels are available, you would not want people to be on Atkins, because their channels would always be trying to suck in glucose and there wouldn't be any glucose there. So it'd be a really bad idea. And then the other, number 16, says, why would you feed them high carbohydrates right before bed? Well, or prolonged exercise, they're going to use a lot of glucose. And so we want to make sure that they have tons of glucose in the blood. So when you do these exercises and you give them an insulin shot, that they are ready to take it in. Okay, so the next questions are dealing with this DNA sequence. So I haven't typed the whole thing, but I've started it for you so you can see what's going on. And so they're saying, if this is the sequence, then number 18 asks, well, what would the other side of the strand look like? So I'm just going to start typing it in here. So remember, A's go to T's. So if I see an A, I write a T. If I see a T, I write an A. And C's go to G's, right? So this is what the, what the opposite side would look like if we were typing it out. Okay, does that make sense? And so on. I'm just going to dot it. And so the other thing that it asks for is in number 19, we're going to label it 5' and 3'. Okay, so let's say that this top one is 5' to 3', then obviously the bottom one would be 3' to 5'. Okay, so they just have to be opposite. Numbers 20 and 21 you can't actually do until we do the next activity, so we can come back to that one. But for 22, we did one of these in class. It says E. coli's DNA is 26% A's, so what percent is G's? So if it's 26% A's, then we know it's also 26% T's. And 26 and 26 adds to 52, which leaves us a 48% that has to be the other G's and C's. So if we just divide that by 2, we get 24% and 24%. Oops. I hope that makes sense. That's just Chargaff's rule. Okay, and that's the end of that activity. So let's jump into the next one.